Again, I'll have a chat to you about a systematic review which we recently published in the BJD regarding reported sequence variants in HS, and also have a brief discussion about what this means for implications for the pathophysiology of what we know of inherited forms that are due to sequence variants in HS. So I'm just briefly going through the breadth of genetic variants there and the critical evaluation of the genetic variants which have been identified there. And I'm going to have a brief talk about the implication of where the location of these polymorphisms are and how this impacts on what we understand of the pathophysiology in these probands with HS. So in terms of the progression of our understanding over the past 20 or 30 years, there are a number of publications early in the 1980s about roughly 1/3 of cases being familial, and then there was the very prominent publication identifying the gamma secretase, the group of genes on the chromosome 1 as being implicated in a family, mostly in Chinese families. And then again, we know there are a number of reported cases which have been identified earlier on today about associated with Darling-Dagos disease, trisomy 21, and then eventually the isolation of gamma secretase in recent years. So overall, the basic pathophysiological mechanisms that are reasonably accepted at the moment are that loss of function mutations in gamma secretase cause deficient notch signaling, which have ongoing pathways, which can also explain the contribution of environmental impacts, which then lead to the manifestations of what we know as HS. However, it's not all what it seems. Gamma secretase, this group of proteins which they're talking about, which have been implicated in early onset Alzheimer's disease, contains 4 separate proteins: nicastrin, presenilin-1, presenilin-2, and anterior pharynx defective 1, which mostly sit in the transmembrane region. However, nicastrin is like a gatekeeper which arches over and protects the cleavage of other sorts of proteins in the extracellular domain. Now, gamma secretase also tends to feed down into what we know as notch, which is down here, and cleaves the notch intracellular domain, which is basically thought to modulate or downregulate notch signaling. And this has been the main process in terms of what we believe is occurring in HS. However, there is a lot going on up here in the extraluminal side of things, which I'll have a little bit of a chat about later. Now, the conflicting evidence that exists is that there are known mutations which occur in probands and families with HS, that cause upregulation in notch signaling. This is completely against what's accepted as being the main pathogenic mechanisms in HS. So not all fits with what we know about the proposition of notch being responsible for these inherited forms of HS. That was the main starting point at which we said, let's have a look at all the different types of variants. So the aims of the systematic review were to have a look at all sequence variants, collate any evidence of protein or functional studies, and critically evaluate them against the American College of Genetics and Medical Genetics Standards and Guidelines. We identified 41 separate sequence variants, which I won't bore you with going through each individually because we'll all be snoring by the end of it. However, it did come across nicastrin, presenilin-1, presenilin-2, and protein serine threonine, PST-PRP1, which is implicated in PASH syndrome. So this is kind of a mutation map of all the different mutations which were identified, and the majority of them were in nicastrin, that overarching kind of protector of the gamma secretase complex, with various smatterings of different other ones as well. Now, in the diagram on the right here, which is in our published article, they're color-coded so that the red variants are truncating proteins, and the green ones are missense mutations, and the blue ones are splice site mutations. The interesting thing about this in the nicastrin, which is the top picture here, is that there's a concentration of splice site mutations near the lid domain, near the large lobe, which is actually the extracellular domain of nicastrin. Now, this is important because it's not where we think it would be if it was going to influence downstream notch signaling. Now, before I go on further with that, I'll go back to the functional studies just briefly. There wasn't too much in the way of functional studies showing that a lot of these polymorphisms are indeed relevant. So there were some mRNA studies. some transcribed protein studies, and a lot of in silico predictive algorithms, which are, a little bit questionable, but only 18 out of 41 proteins identified as having polymorphisms actually had proof in the lab saying, yes, these do affect the transcription of a certain protein. So this is a very busy table, but just an excerpt from the publication showing that, there's a whole different range of mutations here. And look, some of them showed no impact on notch production and notch signaling at all. Some of them showed, a slight decrease in notch signaling here. And some of them only showed a decrease in the production of nicastrin. So basically what it's saying there is that the majority of them didn't show decrease in notch signaling in line with what we understand as the pathophysiology of HS at the moment. And interestingly, a lot of these ones were actually found not only in just familial cases, but also in some sporadic cases as well. However, when we then had a look at whether these were indeed relevant or not, the majority of them were of uncertain significance. So unfortunately, there isn't enough evidence to suggest that a large proportion of these mutations are actually pathogenic without any further work to be done. This kind of emphasises that when we identify mutations now in probands or families with inherited HS, that it's very important that the functional studies are also done as well. Some of the ones which were very strong and using the evidence for pathogenic variants did show that there was likely a pathogenic cause. However, when we had a look at the reasons why they were likely going to be pathogenic, a lot of them were based on the idea that it was affecting downstream notch signaling, but there wasn't actually any proof of that. So we actually did bring into question whether some of these pathogenic mutations were based on the assumption that the disease process is based on intracellular notch signaling, rather than actually having any proof. Now, that kind of ties in to my next point. So, the underlying assumptions is what I found the most interesting part of this systematic review. So, we know that some of these mutations in gamma secretase are associated with HS, not all of them. We know that some of them are only assumed to be pathogenic in HS due to the assumption that notch signaling is the sole pathogenic mechanism in HS. However, it's very, very likely that other loci are likely to be involved, likely through the inflammatory pathways, which Michelle had some excellent pictures of in the last talk. So, the other associated genes are yet to be identified, but I would not be surprised if other types of genes, either through complement or toll-like receptors, are likely going to be implicated in the coming years. Now, the thing that is really important about the nicastrin variants is that the nicastrin variants on the external portion, of the gene here, are involved in the cleavage not just of notch, but in other substrates as well. And a lot of these other substrates, I'll just have a look here, are involved in the binding where nicastrin actually protects the extracellular domain here and is impacted upon the binding of these other proteins here. And these other proteins are kind of using nicastrin as a gatekeeper. So therefore, by having mutations in the extracellular or luminal domain of nicastrin, it's actually preventing this gatekeeper role and having excessive cleavage, or in some mutation aspects, no cleavage at all, of these other proteins which are not involved with notch. Now, when we have a look at the many different proteins that nicastrin can cleave, there are lots. And interestingly, the ones that really pique my interest are IL-6 receptors, IL-1 receptor 1, receptor 2, and also a lot of things like N-cadherin and E-cadherin, which are also involved in the structural integrity of, for example, the lining of the hair follicles. So there's a lot of different things here which may potentially be involved if you would, say, broaden your mindset to think that perhaps if nicastrin is involved in some of these areas, and it's not just notch that is involved, that perhaps there are aberrations in the homeostatic properties of the regulation of insulin-like growth factor, IL-1, IL-6, other toll-like receptors, things like that, that may contribute to the inflammatory mechanisms in HS. So I think there's a lot more work to be done based on the evidence that we can see from some of these mutation studies. So the ones of interest in the substrates other than notch which can be cleaved, as I mentioned, involve the cadherins, other things that are involved in lipids as well as in diabetes, and we know that there's a strong correlation clinically with dyslipidemia, diabetes and other inflammatory mechanisms. So therefore, does this mean that we have to look a bit further and say, could some of the pathogenic mechanisms that are involved in HS, even in some of these familial ones, give us some insight into how HS works in other non-familial cases? So therefore, should we be looking to say that, the insights that we can gain from having a look at these familial cases do lead us to think that perhaps there is another pathogenic mechanisms other than Notch that may contribute to the pathophysiology of HS. So the take-home points from this is basically to say, look, when identifying sequence variants in HS, structure and function is very, very important. And look, taking home the point from this is to question whether indeed the Notch paradigm is the only paradigm that's responsible for HS in some of these inherited conditions, as it seems that there seems to be a dovetailing in terms of some of the inflammatory mechanisms which can be purported from some of these genetic variants, which may be relevant to cases of spontaneous HS as well. So I like to call it the SOTA or substrates other than Notch in terms of future investigations into whether these may be relevant in HS. So thank you very much.